Hi everybody. I am Associate Professor Dr. Ekrem BOLUKBASI at Amasya University in Turkey. My presentation is about especially food additives on the regulation of cancer-related genes in breast cancer cell line. But uh, firstly, I want to thank you so much for the best surgery awards. Thank you so much again. My presentation is about, especially in the introduction, food additives intentionally incorporated into foods are designed to enhance taste, appearance, and shelf life based on the consumer preferences. Concerns arise over their potential health effects, leading to increasing research and regulatory restriction on their usage. The cumulative impact of these additives on human health, even in seemingly safe doses, is the subject of ongoing investigation. Flavor enhancers, including widely used substances like in our research is basis, MSG, MPG, and MDG, are prevalent in a variety of food products. Despite the FDA's classification of these additives as safe, conflicting information and health concerns have created a controversial atmosphere. The conflicting data surrounding the health effect of flavor enhancers, particularly their impact on genes, has led to uncertainty in their safe conception. Especially genotoxicity assessment methods such as in our research, the same real-time PCR is a valuable tool for understanding the genotoxic mechanism of chemical and identifying early molecular biomarkers. Positive results in genotoxicity tests indicate possible direct interaction with DNA, emphasizing the need for true evaluation. In our research and in my presentation, the Stockholm P 450 Stockholm genes, plays a vital role in activating procarcinogens, potentially leading to increasing tumor incidence. BCL2 genes expression analysis provides insight into intrinsic apoptosis pathway, regulating cell death and contributing to cellular genome stability. Changing in the expression of these genes, such as Stockholm P 450 and BCL2, may indicate genotoxic effect and contribute to multi-step process of cancer development. In the current study, the possible toxic effect of MSG, MPG, and MDG substance as food additives, which are frequently used in foods on MCF7 and MCF12A human cell line, were determined by MTT analysis. And then gene expression analyses were performed for the most effective concentration in MTT-assay. Expression of the Stockholm genes and BCL2 genes were evaluated in cultured MCF7 as human-bred adenocarcinoma and MCF12A human mammary epithelial cells cell line treated with MSG, MPG, and MDG for 24 hours. Expression of these genes were provided more information about their genotoxic mechanism. In our research and my presentation, material and methods, cell culture. In this study, MCF7 and MCF12A cell line were cultivated using RPMI 1640, a special name Roswell Park Memorial Institute, or DMEM, Dulbescos modified agile medium, containing the 0.2% sodium bicarbonate as the other components, as you can see in this slide, the other one components. MTT methods, the cell content in a 96 well plate and 100 microliter of RPMA for MCF7 and DMEM for MCF12. A medium were prepared and inoculated into was the microplate was incubated at 37 centigrade and 5% carbon dioxide for 24 hours, and cells were added to surface. Different concentration of exposure, this is important, MSG 27.34 and 437.5 millimolar for MSG, and the other one is here, MPG, MDG, treated with cell for 24 hours. After incubation, MTT solution was added to glass and exposed for 3 hours, and then the reaction was determined by adding DMSO to the glass. Incubated cells were measured with a microplate reader spectrometer at an absorbance volume of 570 nanometer in triplicates. RNA extraction, complementary DNA as cDNA synthesis assay. Total RNA extraction of the control and concentration group were performed according to the RNA plus kit from the Kiagan region, according to suggested procedure by manufacturers. Afterwards, the amounts of purity of RNA were determined using the nanodrop and 1% agarogel electrophoresis. Next, cDNA synthesis was performed using the protocyclic 2-10 cDNA synthesis kit. The qPCR or real-time PCR analysis, the primer of beta-actin as housekeeping gene and cytochrome genes and BCL2 genes used in the study specific to these regions were designed using the information of the MCF7 and MCF12A cell in the gene bank. Information about these genes and the primer sequences were designed for this study are given in table first or 1. Here is table 1 and primers name, length, gene bank number, sequencing, and TM centigrade degree information. Here is, you can see. These are conditions constitute initial denaturation, 10 minutes at 95 centigrade degree at 40 cycles, and the other information is here. You can see the qPCR or real-time PCR analysis containing 3 biological replicates consists of 3 technical replicates using to obtain optimal conditions. In our result and discussion. In the current study, it was aimed to determine the toxicity of MSG, MPG, and MDG, which are frequently preferred flow enhancers in human cell cultures, and to determine the expression of the most effective concentration in terms of cancer-related gene, especially breast cancer cell. The 50% inhibitory concentration as IC50 volume was calculated by determining the MTT results. You can see table 2 upward as possible. The experiment was repeated 3 times for each chemical concentration, and the results are shown in table 2. According to the result, especially IC50 values of MSG, MPG, and MTG were measured as here is red highlighted for MFC7 and MCF12A cells, respectively. In this table, MTT result of MSG, MPG, and MDG treatments of MCF7 and MCF12A cell lines. Here is concentration, MSG 437.50, IC50 value is 227.17 for MCF7, MCF12A, like this you can see in this slide. Real-time PCR especially gene expression, gene related of the cancer as BCL2. We treatment this concentration especially. We use this concentration. The changes in the expression level of the targeted genes in the study were determined by real-time PCR method. The qPCR method is a very effective method for detecting genes, determining their functions, testing, and determining their relationship with various factors. Normalized real-time PCR results of MCF7 and MCF12A cells, and likewise the mean, standard error, and standard deviation of the gene expression data obtained as a result of normalization were calculated, and the results are given in Table 3. The Table 3 mean, standard deviation, and standard error of Stockholm P 450 genes and BCL2 genes expression data obtained as a result of normalization of MCF7 and MCF12A cells, especially MSG, here is concentration and fold change, MPG, concentration, fold change, MDG, concentration, and fold change is here, you can see in the slide. In this graph, MSG and concentration related change in the expression level of Stockholm P450 and BCL2 genes determined in MSG treated MCF7 as breast cancer and MCF12A normal breast epithelium cell lines. Here is the differences. Here is its control group, one fold change, and MCF7 is 2.3 fold change, as you can see here, and MCF12A 0.63 fold change. Here is, as you. This graph is represented MSG results. This slide and this graph is gives information about MPG results, concentration related in the expression level of P450 and BCL2 genes about MPG treated. Here is MCF7 control group, one fold change, especially MCF12A cell line, P450 genes, approximately 5, and as you can here's MPG. Here is 5.12. Here is this graph represents the information about MPG and MCF12A. In this slide, the information about the graphs MDG, the same control groups. BCL2 is bigger than the control, and in MCF12A cell lines, P450 Stockholm genes as here is bigger than the control group. The results obtained from current studies support each other with the literature studies. As a result, the data obtained from this study were evaluated as a whole. The BCL2 gene was expressed more than its normal function against the MSG, MPG, and MDG in MCF7 as spread cancer cells, and it further promoted in scheduled cell proliferation, even seen in cancer cells by increasing the expression level of the BCL gene due to MSG, MPG, and MDG. In MCF12A cells, normal epithelium cells, on the other hand, the BCL2 genes was less expressed than its normal function against the substance and could not fully express its function due to these substances. Similarly, P450 genes as Stockholm genes is less expressed than its normal function against the MSG, MPG, and MDG in MCF7 cell, and it was not enough to eliminate the toxicity caused by these substances. In other words, the MSG, MPG, and MDG worsened the situation of coping with toxicity in cancer cells. And also in MCF12A cells as normal braid cells, the P450 as Stockholm genes was overexpressed against substance than its normal function and was overexpressed to counteract the toxicity from these agents. However, these results need to be supported by using the expression potential of different cancer-related genes with other cell lines. In our study, it has been shown that gene expression analysis is important to understand the mechanism involved in the genotoxicity of this chemical and identify early molecular biomarker associated with food additive-induced carcinogenicity in these cells. In addition, it has been determined that the genes examined in our study are good candidates for future cancer research. Here is the acknowledgement for this research. Amasya University Scientific Research Unit Project No in here is, and collection about MCF7 and MCF12A cell lines. Again, Amasya University Central Research Laboratory cell line collection, we use this. Thank you for listening. Here is my references, you can see. And again, thank you so much for the awards, especially best research awards. Thank you so much again. See you. Thank you so much.